So this video is going to cover sort of the first half, or maybe a third, of investigating populations, because it's quite a big topic, but it's not hard. So we're going to cover sampling, quadrats and transects, abundance, and not biomass, not in this video. But this is stuff that comes up quite a lot in exams, but a lot of it is just sort of remembering like the same answers each time. Like a lot of questions are just sort of common questions, and they've always got the same answer, so it should be easy marks. So the main thing is sort of learning this and then practicing past paper questions. A lot of the time this stuff sort of comes up in question 7, because that's got a lot of data questions. So first thing we're going to look at is sampling. So when we want to study organisms in an environment, we can't study every organism, because if we want to find the average mass of woodlice in a wood, we can't get every woodlouse in the whole wood and measure them. We possibly could, but if we did that, we would be taking a lot of woodlice out of the habitat, and that would cause a lot of damage in the environment. So we'll use samples instead. So we'll study a sample as just sort of a small representative part of a community. And what representative means is the sample needs to represent the population. So it's probably easier to sort of demonstrate this by drawing it. So if we've got a population of woodlice in the wood, so let's say we've got little woodlice and big ones. So let's say in this wood there are 3 little woodlice and 5 big ones. Okay, but let's say that it's a big wood. There are actually 300 little woodlice and 500 big ones. Now if we were to take a sample from this wood, since there are 300 little woodlice and 500 big ones, we could just by chance pick out 10 little woodlice, and we could look at them. We could say the woodlice in this wood are all small, because we've got a sample of 10 woodlice and they're all small. But that's not a representative sample, because not all of the woodlice in the wood are small. They're actually in a ratio of 5:3, where there are more bigger ones than smaller ones. So a representative sample is where if you pick any trait in a population, the representative sample has the same proportion of that trait as the whole population. So if we took a sample of 100 woodlice, I can't draw it, but if we did, we would probably find, if we did it randomly, that we should get them in a ratio of 5 big woodlice to 3 little ones, because there are more big woodlice. So we've got a bigger chance of picking out big woodlice and a smaller chance of picking out the small ones, so we should get them in this ratio, and it should be representative. But what we'll find is, if we take more measurements, then the effective chance is it gets smaller. So if we would take a sample of 10, then we could quite possibly pick out all little woodlice. But if we take a sample of 100, then there's a much smaller chance that we're going to take all little ones. And if we take a sample of 1,000, then there's an even smaller chance. So to get a representative result, we need to take a large sample. And what you should usually say in a question about this is that you must take enough samples to carry out a statistical test. And we're going to look at that again later, but this is just sort of what you need to remember, and you need to be able to write it whenever you get that question, because it's almost always the same answer, exactly the same. It's always similar questions. Another part of sampling is random sampling, and we do this to help get a representative result, because a researcher can be biased. So if you're going to stand in a field and you want to look at the species in that field, there could be nettles, there could be grass, there could be dandelions, but when you're sort of choosing which parts of that field to look at, you might be less likely to choose areas with nettles in, because you don't want to get stung by nettles. even if you don't do it on purpose, you still might accidentally be biased in some way. So we use random sampling, and to do this we use random sets of coordinates. So usually we'll put two tape measures. If we're measuring, say, a field, then we'll put two tape measures perpendicular. We'll put two tape measures out on the field like that, and so they would sort of go up in a grid. It's like 1, 2, 3, so on. And then we would use a computer or any random number generator, but you always have to give an example. So it's probably easiest just to say a computer. So we'd use a computer to generate sets of random coordinates, and then we would take a sample at those coordinates. So if our computer gives us 3 sets of coordinates, then we take samples there, there, and there. So that's sort of it eliminates bias that can be sort of from the researcher, from you when you're doing the research. And this question comes up a lot. This is sort of the part they have to remember. So you'll often get asked, say, count the number of dandelions in a field. How would you go about sampling it randomly? And then you just need to say this. You would need to say that you would form a grid, and you would generate the random sets of coordinates, and you would place quadrats at the coordinates. And we're going to look at quadrats very soon. But again, more results will give you more representative results. But you also have to have a balance, because you've only got a limited amount of time usually. So you have to find a balance between representative and time consuming. So you'll do more about statistical tests in your EMPA, so you don't need to worry too much about what they are and how they work, but you do need to know they have to have enough results to carry out a statistical test. So when you do a statistical test, you'll put all of your results into a big formula, and you'll get a number out of the formula, and that number is P. And P is the probability that results are due to chance. So when we were saying before, if you took the woodlice out of the wood and you picked out 10 woodlice just due to chance, that's sort of what this means. We use a statistical test to calculate the probability that they are due to chance. And the lower the probability that are due to chance, then obviously the higher probability that they aren't due to chance. And in biology, we usually say that results are valid if the probability is less than 0.05 or 5%. If the probability is greater than that, then it's too high, because they could be due to chance, so the results are invalid and we just ignore them. You get asked this a lot, and you just need to remember exactly this. So if P is less than 0.05, the probability that results are due to chance is less than 0.05. So we use quadrats for measuring the abundance or distribution of plant life, and we're going to look at abundance again later, but distribution just means sort of how they're spread out. So if you've got a muddy field and you've got some plants, they are not evenly distributed. They're sort of spread out, and the percentage of the field that they cover is quite small. So distribution just tells us sort of about the way in which all the life is spread out throughout the environment. But abundance is a measure of numbers, so the size of a population, how many there are. Now, frame quadrat is a square frame, just like this, and is sometimes sort of divided into a grid, but not always. And you put this square frame down. If we go back to this, then we'll put our square frames down here, here, and here. So we'll put the square frame down at the coordinates, and we'll look at all of the organisms inside the frame. So if we want to find abundance, then we would count the organisms inside the frame. If we wanted to find the mass of organisms, then we would pick all of the organisms out of the frame and go and measure their mass. So that's what we do with a frame quadrat. A point quadrat is is a little thing like this, which we put down on the ground, and it has holes in the top like this, and it has a needle, and we drop the needle through each hole. That's not a very good needle, but you probably get the idea. You just put the needle through the hole, and whatever plant or whatever insect or whatever it lands on, then you look at that. So if this was landing on a dandelion, then you would count a dandelion, and if this was landing on grass, then you would count grass, and so on. So this is sort of, it's less arbitrary than a frame quadrat, because with a frame quadrat, if you're going to count the organisms, you can sort of, if you've got grass, you sort of have to guess the percentage cover, whereas if you wanted to guess the percentage cover from a point quadrat, well, if you had 5 holes, then 2 of them were landed on grass, then you know that 2 out of 5 points are on grass, and then you could work out that as a percentage. So 2 over 5 times 100 equals 40%. Whereas with frame quadrat, you would just have to guess the percentage cover. So if you had grass here, 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 then you could sort of guess that that's 50% covered in grass, but you don't know for sure, and you've got no real way of sort of making it standard, because a different researcher might say that that's not 50%, that's something else. Now the size of a quadrat that we use, it depends on the size of the species that we're going to study. So we'll usually do a preliminary test to determine what size of quadrat to use, and we'll plot a graph. So we'll place quadrats down, like a certain number of quadrats of each size. So small, medium, and large. Obviously, you'd have numbers, so you'd have like maybe 1 meter, 2 meters, 3 meters, and then we'll plot this graph of the size of the quadrat on the bottom with the number of species, the number of different species along the side. And then we'll plot this graph to get sort of a curve. And what we want to do is find the quadrat that collects all of the species available, so we're not going to miss out any species when we're measuring. But we also need a balance, because if we use a quadrat that's too big, then it's going to take up a lot of time, and it's not going to be worth doing. So we need to sort of find a balance. And in this case, we'd use the medium quadrat, because at this point it's getting all of the species. And the large quadrat, that's getting all the species too, but there's no need for it because it's bigger, when this one's doing exactly the same thing. So if a species occurs in groups, smaller quadrats are going to give more representative results. So for example, if we've got field, big field, and we've got, say, clover dotted around the field in little groups, if we want to put quadrats down and measure the abundance of clover, I'm not going to come to abundance again later, but if we want to put quadrats down and measure the abundance of clover, we might put, if we're using big ones, then we might do that and that. Now, one measure of abundance is frequency. Frequency is how often the species that you're looking at appears in a quadrat. So in this case, it's 1, because it's 100%, because it's in this quadrat a tiny bit, and it's in this quadrat. If we put another quadrat down, then it might not be, but then we put another one down, and it's in that one, so this is 75%. But if we look at it, we can see really clearly that the clover is not spread over 75% of this field. There isn't that much clover in the field. So that's not going to be representative at all of the population. But if we use smaller quadrats, the clover is only in one quadrat here, so that is going to give us a more representative result. Transects. A transect is basically a line. So you put a line along the ground like this, and you would look, well, you would pick a point on the line, so you might randomly generate a number, say 5, so you'd go to 5 on the line, and you'd look at any species that's under this point on the line, and that's the species that you're looking at. So if you're counting abundance, then you count the species under there. We usually use systematic sampling, which we'll talk about soon, but another type of quadrat is a belt transect. So instead of one line, we would have 2 parallel lines along the ground, and we would pick a point on the line, and we would look at all the species in the belt between these 2 lines, because this is the belt here. The belt's just the area between the 2 lines, and we'll look at the areas in the belt. Now we usually use a transect when a transition occurs in the environment, rather than for studying abundance. So we use it for maybe distribution. So if we've got an environment like a shore, a rocky shore, we've got sea here, and then we're going to have different species living at different heights up the shore. So here I might have seaweed, crabs, and here I might have reeds. I don't know if you get reeds on the shore. And here we might have trees. And we might want to look at the distribution, or sort of how the distribution changes of species as we go up the shore. So we put a transect line down going up the shore, and we would look at the species on the shore at different points on this transect. So we would look here, and we would see maybe at 5 meters below the sea level we've got seaweed and crabs, and then we would look here and see exactly at sea level we've got reeds, and then we would look here, and then maybe 10 meters above sea level we've got trees. So we would use this to sort of see how the distribution of species change as a transition occurs through the environment. We could also use it sort of in maybe a salt marsh, and this is sort of the same situations. You would have the water and a shore, and the species are going to change as you get away from the water. Now we usually use systematic sampling rather than random sampling, because we want to look at how the distribution changes at different distances rather than get a representative sample, because there's no point in getting a representative sample of this, because nothing random is going to represent this because it changes. So if we were to pick random numbers, they could all be up here, and we'd be looking at trees and maybe squirrels, but then we're not going to see anything down here. So it's sort of pointless using random sampling in this situation. We use systematic sampling. Now systematic sampling is just when we use sort of a method for determining where to sample. So for example, we might say that we're going to take a measurement of the species present at every 5 meters. So we take a measurement here, here, and here, and it's always going to be even, and we're going to take it at a set distance, and we're going to see how every 5 meters the distribution changes. And finally, we're going to talk about abundance. Now abundance is basically the number of organisms in an area, but in a lot of cases we're not going to count the number of organisms in an area because it's impossible. Like if we put a quadrat down on the grass, we can't count every single blade of grass in the quadrat. It's going to take forever. But instead of that, we can estimate the frequency, and this is what we looked at before, where we had the clover in the field, and the frequency was how many times the clover was inside the quadrats. So the species is the likelihood that a species will occur in a quadrat. So if we've got clover in a field again, and we put 3 quadrats down, and clover appears in 2 of them, the frequency is 2 out of 3, which is 0.6 occurring. And that is the chance that it will occur in a quadrat. Percentage cover is an estimate of the area in each quadrat covered by a species. So this is a better measurement, because as we saw before, if you put quadrats down, then if you get a single tiny percentage cover of that species in a quadrat with frequency, then count that as one quadrat, and that sort of gives an unrepresentative view, because you could have 1% of the area of a quadrat covered by a species, but we only know in the results that the quadrat had that species inside it. Percentage cover is better measurement, but it's not perfect. So if we had our clover again, we put a quadrat down here, we would estimate that this is about 50%. You'll make the measurement to the nearest 5%. So if you've got a quadrat here, then you might say that that's 5% or 10%, but you would never be more specific than that, because you can't really estimate it that specifically. And finally, we calculate the mean of all the samples collected, and this is to get an average, so we can get one single result to give us an idea of how many organisms there are. So to calculate the mean, you add all the separate results together. So let's say that these are the results that we got from our 3 quadrats. You add them all together, and then you divide them by however many results there are. So there are 3 results here, and then you get the mean, and that is sort of what you would state as the abundance for the whole field. So this was just part 1. In the next video we're going to look at mark release recapture, which is what you use for sampling animals rather than plants, because animals can move around. So while you could use a quadrat for wood lice, you couldn't use it for bigger things. You couldn't use it for faster moving things because you just couldn't count them. And we'll also look at ethics in biology.